I would like to thank the organizers for this opportunity. I shall start my presentation. So I start by wishing you all a very good afternoon. The title of the talk is The Amazing World of Nano Devices. I suppose you can all see the mouse on the screen?
Yes.
Okay, fine. This is, of course, my name and my affiliation. And as I said, I would like to thank the organizers for giving me this opportunity to talk to you. And here is my address. Actually, this is my address in the previous place where I was working in the Institute of Science. It is still active, so if you need to look at anything regarding me, of course, you can look at this site. It is an outline of my talk, but interestingly, you see, normally you put acknowledgements at the end, but I thought I shall start with acknowledgements in case I run out of time, but I have been given 2 hours, so therefore I may not run out of time. Again, I shall describe to you the aim of the talk and then give certain apologies. You may wonder why apologies. Well, I will tell you the reason. You will see when you hear my apologies. Then I shall speak a little bit about motors. Motors seem to be very familiar things. What is there to talk about motors? Well, you will see. Then I shall talk about the passage of a DNA molecule. I suppose the science background of the listeners is quite good, so that I don't have to explain to you what a DNA is and so on. So we will have a long molecule like DNA passing through a nano-sized spore. Why worry about this? Well, in fact, many of the things that I talk about are topics on which I have done some research, even though I will not go into the details of my own work. So then you have a DNA passing through a nano-sized spore. Then I shall talk a little bit about a very interesting nano-sized system, which is ATP synthase. And then I shall talk about viral packaging. Well, these days everyone should be interested in viruses. What I am going to talk about is actually viral packaging, and then talk about nano devices. And ask what about the future. Now you see, the thing is, if you look at my PowerPoint, my presentation, you will find that I have taken characters from Tintin comics, which is not really legally allowed. Anyway, they are there in my slides. And I have taken some animations from the internet, but whenever I have done it, I have given credit. So the aim of the talk is actually to make you all aware of the engineering marvels that exist in nature. There are absolutely marvelous things existing in nature, of which you have no idea, right? Well, maybe if you are a biologist, you will know, but others usually had no idea. And in fact, I shall also, these are actually in the nano world. And in fact, I shall talk about the scientists' attempt to imitate them. And here are my apologies. You see, my own area of expertise is actually chemistry. Even in chemistry, you see, if you say this is the whole of chemistry, my knowledge is very limited. I do have some interest in physics, because many of the things that I do, a physicist will classify it as physics. And I have almost no idea of biology. See, the thing is, the way I work, I get into new areas, but then it is not necessary that I should know everything about that new area. And in fact, I try to manage with whatever knowledge that I have of physics and chemistry. So therefore, my expertise actually is in this yellow region, perhaps a little bit. And the talk today actually is in that yellow area. The area is biophysical chemistry. And as far as biology is concerned, I have no great knowledge. I mean, you can, even a high school student can ask me simple questions in biology and I wouldn't know the answer. But as far as the theory of this is, many of the things that I'm going to talk about are concerned, I have done research on them. Now let me look at this question. What is nano? Well, this is only a reminder, because I'm sure all of you these days would know what nano is. Nano means 10 to the power of -9. Okay, nano is simply another word for saying that, okay, it's 10 to the power of minus 9 something. Now, what is the size of a hydrogen molecule? That person would find difficult to answer. But then so what is the size of a hydrogen molecule? Well, it's about an angstrom, 1 angstrom. How much is 1 angstrom? If you are old like me, then it is actually 10 to the power of minus 8 centimeters. Okay. Now, another way of saying the same thing is actually it is 10 to the power of minus, 0.1 nanometers. Now I ask the question, what is a motor? Well, the answer is that a motor is something that will consume chemical and does work. Then, of course, you may say that, okay, that doesn't include electrical motors, which is quite true. But if you think of the petrol car, then of course the petrol car actually consumes petrol, and there is a motor that runs on petrol. So that will be my definition. A motor is something that actually consumes some chemical and does work. So for example, if you had a big ship like this one, then you see this is rather huge, and of course a ship will have several motors, not just one motor inside it. Many motors would be there. But then these are all very big motors. Now the question that I ask is, how big is the smallest known motor? How big is the smallest known motor? You think about it. What is the smallest size that you can think of a motor having? And again, if I showed you this picture, what would you, and asked you, what is this? Well, you see, if you are not familiar with it, then you will say it looks like a flower, but it doesn't really look like a flower from the picture. If you have showed it to a kid, of course, he will say that it is a flower, but it is not really a flower. Then what is it? It is actually a motor. So what motor is this? Before I go into the details of this motor, the height of this is actually 15 nanometers. 15 nanometers means it is 150 angstroms in height, 150 angstroms. Then the width of this is, the width is actually 10 nanometers, that is about 100 angstroms. So where does it occur? This is something that occurs in nature. In fact, it occurs in the cell inside your body. It is there, this nanomotor. And where is it? Well, this is a schematic picture of the cell. Or if you think of a plant cell or any cell, you will have some structure like this. It will have a nucleus and so on. What is of interest to us is actually this mitochondrion, which is sitting there, which is referred to as the powerhouse of the cell. So if you took this mitochondrion alone and had an expanded view of it, this is how it is going to look like. It has, if you look at it, you see it has a covering consisting of 2 membranes, and there is a region in between these 2 membranes, which is shown in green color here. What I am going to do, and that region I shall refer to as the intermembrane space. And what I am going to do is I am going to take this portion enclosed within this blue rectangle and magnify it. Oh, before I do that, I would like to tell you that this intermembrane region is having a high concentration of hydrogen ions. So in layman's language, that means that this is acidic. This region is more acidic in comparison with the outside, which means that it has more protons or more hydrogen ions. So therefore, you see, if you think about it, you can say there is a gradient. You see, from the inside is richer in protons than from the outside. And how is that gradient of protons maintained? The energy from photosynthesis is used to keep that gradient. So therefore, schematically again, you can say that the intermembrane region has a more positive charge. That's another way of saying it. I mean, it's not really positive charge located there, but you have more protons. Okay. So these protons, you see, suppose there is a hole in the membrane. Suppose there is a hole in the in this membrane that I'm showing with the mouse, then what will happen? These protons will actually leak through, because you see here in this region, the concentration of protons is lower, and therefore these protons will pass through the hole into that region. And the way that is happening is shown here, right? From the green region, you can see there are protons there. This is rich in protons or hydrogen ions, and these protons will actually pass through that hole and go to the other side. This is essentially driven by the concentration gradient, because there is a difference in concentration. Protons which are in the region where they are high in concentration will go to the region where they are low in concentration. This is actually, a chemist will say that it is driven by the chemical potential. So then you ask, well, if that is happening, do you have actually holes in the membrane? The answer is yes, there are holes. And these holes are actually in an enzyme which is sitting in the membrane. And this enzyme is actually called ATP synthase. Why is it called ATP synthase? Because it synthesizes the molecule known as ATP. So interestingly, what happens is that the passage of the protons through this hole causes the formation of ATP. So how is ATP formed? Well, there is ADP, the molecule called adenosine diphosphate in the system, in here. And this adenosine diphosphate and phosphate, they exist there. And what happens is that the enzyme takes them, makes them react to one another, and that results in the formation of adenosine triphosphate, which is ATP. We will see more details about this. But the interesting thing is that this object, this whole thing is only nanometers in size. As I said, maybe 15 nanometers high, 10 nanometers wide. And in fact, it is a rotary motor. That means it is rotating. Why? How does it rotate? In fact, it is one of the most powerful known motors. And of course, if the moment the human beings become aware of such a thing, they will ask, well, can we make use of them to do useful things for us? And the answer is actually yes, we can actually make use of them. But before again I go into that, we need a little bit of background. Well, the background, let me just remind you, this would be familiar to most of you definitely. If I asked you a question like, what is the biggest molecule that you know of? What would be your answer? Well, you can think about it. In a classroom, I would normally ask the students and wait for the answer. But because we are doing this through video conferencing, I am not going to wait. The biggest molecule that we know of are actually DNA. So how big can DNA be is the question. Well, DNA is actually standing for deoxyribonucleic acid. They can be very long. They can be as long as 3 meters in length, which is really macroscopic in size. But even though they are very long, their thickness, you see, this is like a string which is 3 meters long, but the thickness is only in angstroms level. The thickness is only 20 angstroms. Okay. So therefore, if you say that this is, again to give you an idea, if you say this is a DNA molecule and I'm going to magnify it, suppose I magnify it, how many times will I magnify? Let us say 10 to the power of 6 times. Then you will get such an object if you can magnify it. And this object will now be 300 kilometers long, but only 2 millimeters in width. So it is like a very, very, very long string, but very thin. So that is how the DNA is. You see, it is very long usually, and it is very thin. Now, what is so surprising about that? Well, the answer is, well, again, going into the details of the DNA molecule itself, this is a schematic representation of the DNA. It is inside the cell, it is inside the nucleus actually. and it is double-stranded. These are things that you normally would know of. And there are these bases which are called A, C, G, T, and so on. We will come to them. And well, what are these bases? Adenine, guanine, cytosine, and thymine. Well, these are the bases. We will not go into the details of this as far as we are concerned. We just know that there are different bases sitting there. The one is adenine, which is denoted as A. Next is guanine. The next is cytosine, and the fourth one is thymine. So you see, in the double-stranded DNA molecule, this is the double-stranded DNA that I have shown here. You have A, T, A, T. These are actually the bases. They are arranged in some order. And here in this portion, I have separated the two strands. Usually these two strands are held together by hydrogen bonding, but if you are able to break the hydrogen bonding, then you will get what is referred to as a single-stranded DNA. So this is a single-stranded DNA, and it has a particular sequence. You see here, for example, the sequence is A, C, G, A, T, etc. So when you say you are sequencing the DNA, you are actually saying that, okay, you will find out the order in which these bases appear. A, for example, here it is, this is a particular sequence, you say. So it will be A, C, G. This is the same thing that is drawn there, So therefore, you will have A, C, G, etc. This is what you refer to when you say that you are sequencing the DNA. So this is the most important thing as far as the DNA molecule is concerned. It is the sequence of the bases that is the most important. And the other interesting background material that I would like to tell you is that, you see, these days, it is actually possible to do experiments with single molecules. You see, in the early days, when I was a student, you see, you will do an experiment in the lab, and that will have millions and millions of molecules in the solution. If I take some material inside a test tube, that definitely has millions and millions of molecules. But these days, it is actually possible to do experiments with single molecules. So what about these experiments? Well, if you had told me this, let us say 25 years ago, I would have laughed at you, because I would have thought that that is impossible. But now it is possible. 25, or maybe I should say not 25, but 35 years ago. So in these days, it is possible to do single molecule experiments. So let me tell you of one such single molecule experiment, which is actually very nice and useful. Why is it useful? Because it has been used to sequence the DNA. For application of this experiment has been made to sequence the DNA. So what is the experiment? Well, this is in principle is a simple experiment which can be done in an ordinary lab. You say that you have a potassium chloride solution. This is potassium chloride solution, and you have a membrane separating two parts of this solution, right? The left-hand side as well as the right-hand side, they are separated by a membrane. The membrane has a single pore. This is the important thing. It has a one pore. And that pore has a size of 2.6 nanometers. 2.6 nanometers is actually 26 angstroms. So that is extremely small. Even though it is extremely small, this pore is wide enough for a single-stranded DNA to pass through. Because single-stranded DNA, as you have seen, double-stranded DNA is only about 2 angstroms, so single-stranded DNA, not 2 angstroms, but 2 nanometers. So single-stranded DNA will be about 1 nanometer in diameter, and therefore that can easily pass through this nano-sized pore. Now, what you have to imagine is that you put DNA molecules on one side of this system, and you apply a potential difference, right? You apply a potential difference between the left-hand side and the right-hand side. DNA molecules would have, if the pH is correct, the DNA molecule would have a negative charge. And the DNA, you see, because you apply this is your positive electrode, what will happen is that the molecules will try to go from the left-hand compartment to the right-hand compartment. And there is only one way in which they can go. They have to go through this nanopore. So therefore, you can actually force this single molecule, just one molecule. You see, this pore is very thin, and therefore only one molecule can pass at a time, not more than one. So when you apply this potential difference, what is going to happen is that the molecule, DNA molecule, only one of them, even though you have several hundreds or maybe thousands of DNA molecules on this side, only one single stranded DNA molecule will pass from the left-hand side to the right-hand side. And you can see that, I suppose, as an animation. Let us see. So this is the animation. So this is a long DNA molecule. It has passed from the left-hand side to the right-hand side. Now, how do we know that the DNA has passed through? That is an interesting question. And for that, actually, you can measure the conductivity of the system. So when you apply potential difference between the left-hand side and the right-hand side, you see there are potassium ions and chloride ions in the medium. These ions will actually pass through the pore. And so therefore there will be a current that is passing through the system, and current is something that we can measure very accurately. So therefore, when the pore is empty, you see you will have a current passing through the system, which will be large. But then when DNA is passing through, you see part of the pore is obstructed, and therefore there is not space available, as a result of which the current of ions passing through will reduce. And therefore, when the DNA actually is in the pore, what will happen? You will see a lower current, and that is what is shown in this animation. Let me play that animation once more. You look at the current now. The current was having this value, but when the DNA is entering, the current has dropped to that value, and therefore you are actually able to measure precisely how long the molecule has been sitting in the pore. How long does it take to pass through the pore? You can measure that by simply measuring the current. Let me play that animation once more. You look at the current now. When the DNA has entered the pore, the current has actually dropped. And when the DNA has passed through fully, the current is back to the original value, and therefore you are able to measure experimentally the amount of time that it takes for a single DNA molecule to pass from the left-hand side to the right-hand side through the pore. And that itself is interesting because this is an experiment done with only one molecule. But then, again, you think about it. What is the use of this? Well, the answer is that, you see, we are interested in determining the sequence of the bases in this single-stranded DNA. Let me just go back. Yeah, well, you see, you look at this. This is the sequence that you have. Now, interestingly, you see, only one of the strands is actually passing through, because I have taken single-stranded DNA to start with. I have not taken double-stranded DNA in my compartments. So therefore, you don't have double-stranded DNA, but you have single-stranded DNA. And if you look at a particular strand, let us say you have this strand. Then you have sequence like A, C, G, T, etc. The interesting thing is that, you see, the size of this A, at base A, is different from the size of C, and that is different from the size of G. So imagine that you have the base A sitting inside the pore. Then the blockage will be larger in comparison with the blockage caused by a C. And it will be perhaps different from the blockage that is caused by G. And it will be different from the blockage caused by T. And therefore, if you look at the variations in current, at least in principle, right, it is possible to know what is the base that is passing through the pore. Let me repeat. The bases have different sizes. Depending upon the size, you see the current is going to be different if an A is sitting, or when a C is sitting, or when a G is sitting, or when a T is sitting. And therefore, by looking at these small variations in the current, it should be possible for me or for an experimentalist to identify the sequence of the DNA. And therefore, by looking at these small variations in the current, people thought it is possible to sequence the DNA, but this turned out to be a simplistic thing because the pore is actually long, it is not small. The pore would have a length of maybe 100 angstroms. And if it has a length of 100 angstroms, you see what happens is that at any time there are 30 bases sitting inside the pore, and therefore you will never get single base sensitivity. So therefore, you see, it seemed a little bit far-fetched when this idea was proposed. This idea was proposed in 1996. So these are all latest things I see. I mean, this is of course only about 25 years old. The idea itself. What is the problem? The problem is actually the pore is quite long. At any one time, you see there is something like 30 bases sitting inside the pore, and therefore it is impossible to have single base sensitivity. And how do you attain single base sensitivity? You have to reduce the length of the pore. What should you reduce it to? Well, you should reduce it to such a level that there is only one base sitting in the pore at any given time, or one base blocking the current, and the other should not be blocking the current. Then you will be able to attain single base sensitivity. So let me go back to the slide that I was talking about. So then you also have this other question. Well, it is nice to say that have a nanopore and then have this experiment, but how will you make a nanopore? That is another interesting problem. And that is not an easy thing. Well, interestingly, you see, it is difficult for a chemist or a physicist would have to do very sophisticated experiments to make a nanopore. These days, people have succeeded. But even in 1996, you see, it was possible to have a nanopore. How did they make the nanopore? Well, you use your knowledge of biology. There is a protein which is called alpha hemolysin. I'm going to talk a little bit about alpha hemolysin. It is a protein that self-assembles. Self-assembles means, you see, if you have these molecules in a medium, they can assemble. These proteins can come together, assembled together and have an assembly. And the interesting thing about the assembly is that it has a nanopore. It has a nano-sized pore in that assembly. You will see the assembly in a second. And in fact, this is secreted. This alpha hemolysin is secreted by an organism called Staphylococcus aureus. Okay, that's a biological name. I don't know what it means. Anyway, this is alpha hemolysin. This is the assembly that I'm talking about. You are seeing a cross section of the assembly. And the assembly has a nice property that there is a pore. So what actually happens is that this organism, Staphylococcus aureus, what it does is, if it comes near a cell, it secretes this alpha hemolysin. Alpha hemolysin will go into the cell membrane of the cell near which it is sitting and then insert itself into the membrane, and then it will assemble so that you have a nano-sized pore created in the cell. Why does the organism want to do that? Well, once this pore is created, you see the chemicals from inside the cell can come out, and the organism wants those chemicals. So this is a little bit of details about alpha hemolysin. It is a protein that actually kills. This is the Staphylococcus aureus is a common cause of infection in humans. It secretes a toxin known as alpha hemolysin that kills human cells. Well, it is going too fast. Let me just stop it and go back. So a toxin that is known as alpha hemolysin that kills human cells by forming holes in their membranes, through which the chemicals essential to the survival leak out. This movie illustrates the structure of the hole, which is created when identical proteins released by the bacterium, which are shown in different colors, as you will see, assemble in groups of 7. So therefore, 7 such protein molecules assembled form this assembly, which has a pore, and the result is a hole in the cell membrane. You will see the assembly. So this is the assembly. Each protein is shown in a different color, and you can see that there is a nice pore. Okay. In fact, then what you can do is, you see, imagine you have such a nano-sized pore, and imagine you have a DNA molecule on one side, single-stranded DNA, and then it is possible for you to force the double-stranded DNA to pass, sorry, not double-stranded, but single-stranded DNA to pass through this nano-sized pore. And this is an animation. The string that you see passing through is actually a DNA, single-stranded DNA molecule, and it will go from one side to the other side. And this, of course, will result in a blockage of the nanopore, and that will result in a dropping of the current, which you can measure experiment. Well, these days, actually, people have fabricated nano-sized pores. You see, for example, this is a pore which is much wider, 90 nanometers, but artificially fabricated. And here you see you have regions where the single-stranded DNA can sit comfortably. It can sit here or there. But in this region, you see the DNA is not comfortable. So therefore, you will have to force the DNA by application of a potential difference to move from this to the other. And such experiments have already been done, actually. And you just need to apply an electric field causing the movement of the single-stranded DNA. So this is a question on which I have done some research, actually. See, for example, you can ask a question, how much time will it take for a molecule to go, a long molecule to go from one side of the membrane to the other side? This time, you can measure it as a function of the length of the molecule, and obviously if you increase the length of the molecule, the time will increase. But how does it increase? Would it increase like the square of the length, or would it increase like the length of the molecule itself? There was also a suggestion that it goes exponentially, which actually means that the time will be much, much longer. So these are the areas on which I have worked on, though I will not go into the details. Actually, the time is actually proportional to the length of the molecule. Essentially, I mean, that is a simple idea. If you make the molecule longer, it will simply take longer time. Now, this has actually been used to sequence the DNA. You see, I told you that it seems difficult because the pores are actually very long, and therefore you have a difficulty. And it is difficult to attain single base sensitivity, but people have successfully done this. And how did they do it? They made the artificially this is where the power of the chemist comes in. They made the pores, the effective length of the pore smaller. How did they make that? Well, I will tell you. I will not go into the details, but I will tell you how. Okay. Yeah, well, what you are going to see is this animation where you have a double-stranded DNA. You have a double-stranded DNA here. This is actually an enzyme which will cause the two strands to separate. You see, you know that the DNA has to replicate, make copies of itself. There are enzymes which will actually cause the two strands to separate, because if you want to replicate a DNA, you need the single strand. So therefore, there are enzymes which cause the two strands to separate, and therefore this enzyme, what it does is, it causes the separation between the two strands. And then, so therefore, you have only one strand shown here, and that strand is actually going through the nanopore. Okay. So here you have the nanopore, and this is the one you are going to apply a potential difference between the two sides of the nanopore and measure the current. And when the different bases are inside, you see, you will have different extents to which the pore is blocked, and therefore the current will be different. But still, when you look at the length of this pore, you can see that it is quite long, and at any time it appears as if there are several bases, something like 30 bases would be inside this region, this nanopore. Anyway, let us look at the experiment. Let us see. Okay, now we look at it in more detail. You see here, if you look at the experiment, you see this is a single strand that is passing through. But if you look at this part of the animation, you will see that this is a cross-sectional view of the pore. you see that there are protrusions here. These protrusions, there is one here and there is another one there. So therefore, the effective cross-section of the pore in this region has been reduced. Well, how does one do that? Well, this has to be done chemically. So that is where the ability of a chemist comes in. He can actually do some chemical modifications of the pore so that in this region you have a smaller cross section of the pore, and therefore you see that the region is very small, and that region is only about 3 angstroms in length, and therefore at any time you see passing through that region there will be only, okay, so therefore you see you have one base passing through this region at any given time, and therefore the current that is passing through the system will depend upon which base is passing through the system. So that is how it is. And now I shall pay the place. And then, so therefore, when these bases are passing through, the currents are going to be different. So you can see here the currents are actually different. I shall play the entire animation once more once this is done. So you can say that, okay, that probably is an A, this is a C, this is a G, and that is a T perhaps, and so on. So therefore, by measuring the current, you see, this is not a very complex setup. This is a fairly simple setup, and using that, you can actually, don't worry about this portion because I'm not going to talk about this portion. Now, let me play the animation once more. Let me again stop here. Well, you see, when you look at this, you see, it seems very simple. You have to have this enzyme which separates the two strands, and then that will cause the formation of a single stranded DNA. That single strand will pass through. But these don't occur spontaneously, which means that you have to spend energy. So where does it get the energy to move, for example? And that, of course, the enzyme provides. You see, enzyme consumes ATP. That gives the enzyme energy, and that is driving the single strand through the pore. So this is a very nice example of the power of nanotechnology. So therefore, you realize that, you see, here is a very nice experiment which can be used to sequence the DNA. And as I said, in 1996, when I saw this proposal, I thought this is impossible. But then, of course, people who are very clever did do this experiment. So this, for example, is an experimentally measured current from an actual equipment which was fabricated. And using this, it is possible to sequence the DNA. So I said this is the people have fabricated an equipment and it is available. This is made by a company. I have no interest in the company, and this is not an advertisement for the company, but a company called Oxford Nanopore has made this. This is the device. You look at it, you see this. When you look at a figure, you don't get an idea of how big it is. Actually, it is a device that can be held in your hand. So it is that small. The cost is about $500, I'm told. At least it was $500 when some 4 or 5 years ago. Now the price may be higher or maybe lower, I do not know. And made by Oxford Nanopore. So therefore, this device can sequence the DNA. And in fact, this is the sequence that was determined by that company. This is this slide was prepared quite some time ago. So I have written today. But that means it would have been a 3, 4, or 5 years ago. And the time that it took the molecule to pass through the pore was only 273 seconds. And in those 273 seconds, you were able to determine the sequence of that particular DNA, which reads like this. So that is the sequence. So therefore, you realize how simple it is using this nanopores. And this is the actual current that was seen in the experiment. So with that, you see, I hope I have convinced you that nanotechnology can be very powerful. In fact, we human beings can manipulate things and make use of things that are available at the nano level. Now let us go back to the ATP synthase. So this is ATP synthase. So what is ATP? ATP is actually a molecule which is made of ADP. This is a schematic representation of ADP. ADP has 2 phosphate units. So one adenosine and then 2 phosphate units. So that is why it is called adenosine diphosphate. And then you have phosphate, which is an inorganic material. And these two can combine together and you will get an ATP, adenosine triphosphate. It is a triphosphate because there are 3 phosphate units. And this molecule is known as the powerhouse of the cell because all the chemical energy is stored in there. And if you hydrolyze this molecule, energy is released, and that energy is the one that is used to do all the things that we are doing.
Concentration gradients are a key component of the biological world. The potential energy from these gradients is often used to perform biological work. Here we will focus on hydrogen ion concentration gradients. Hydrogen ions are also known as protons. A gradient exists when there is a higher concentration of a molecule in one compartment compared to a neighboring compartment. This animation will demonstrate how the potential energy that results from a hydrogen ion gradient uses ADP and inorganic phosphate, also known as PI, to synthesize ATP. This process involves an enzyme complex called ATP synthase. Gradients and the potential energy they create are key aspects of the biological world. A good example of the use of a gradient occurs in the mitochondria when ATP is synthesized. ATP is synthesized by ATP synthase, a large complex of membrane-bound protein. Here we see ATP synthase, along with other membrane-bound proteins. Notice the large difference in the number of hydrogen ions on the 2 sides of the membrane. This difference is a hydrogen ion, or proton, concentration gradient. The energy associated with this gradient is used to synthesize ATP from ADP and PI. This occurs at the ATP synthase complex. One hydrogen ion enters the ATP synthase complex from the intermembrane space, and a second hydrogen ion leaves it on the matrix space. The upper part of the ATP synthase complex rotates when a new hydrogen ion enters. Once 3 protons have entered the matrix space, there is enough energy in the ATP synthase complex to synthesize 1 ATP. In this way, the energy in the hydrogen ion gradient is used to make ATP.
Okay, I shall temporarily stop here. The reason is that you see, the idea that here is that you have a concentration gradient of protons, and therefore the protons will go from one side to the other side. And you will notice that when a particular proton is passing through, that actually causes a part of the system to rotate. Now these are nanomotors. You see, their size is only 10 nanometers, as I told you, and they rotate because of the passage of the proton. And this rotational motion, we believe, I mean, there is no proof yet, it will mechanically force the adenosine diphosphate, ADP, and the phosphate molecule to come together. You are forcing the 2 molecules together, and that is what this rotational motion does. You force them together, and then when they are mechanically brought together, they undergo a chemical reaction. So this is an example of a new branch of chemistry, which is referred to as mechanochemistry. You cause a chemical reaction to occur using mechanical force, and that is what this enzyme does.
Now let's watch the process again. Notice how the proton enters the ATP synthase and exits into the matrix space. Once 3 more hydrogen ions have crossed the membrane, another molecule of ATP will be made. In this example, the hydrogen ion gradient is large enough to produce 6 ATP molecules. Please watch as the remaining ATP molecules are synthesized. The process has now completed, and the result is an equal number of protons on each side of the inner membrane. Without a gradient, there is no more energy available to make ATP. In biological systems, however, a gradient is always maintained. The mitochondrial hydrogen ion gradient is generated as electrons pass through 3 membrane complexes. That process can be seen in the mitochondrial electron transport chain animation.
Okay, this is another animation, but it is not as interesting as the previous one. But anyway, this is actually an animation which shows the rotation in. Okay, so therefore we have seen that there are rotary motors which do mechanochemistry. That means you force 2 molecules to come together and cause them to react. So these are referred to as rotary motors, but you also have motors which are able to walk, proteins which are able to walk, something like the walking of this person, and you are going to see one such in the next slide. Well, then of course you will ask, why does the molecule walk? This again is happening inside the cell. In fact, it is walking because it is acting like a coolie at the railway station. You will see what it does. So this is the, if you look here, you see this is the thing that is going to walk. And if you look at the figure or the video, you will see that there is a huge bag. This is the baggage that this is the coolie and this is the bag, and he is going to carry that bag from one end of the cell to the other end. It is walking along filaments. These filaments are known as the actin filaments. So let us now look at the animation. As the animation starts, you see this nano-sized walker. He will start walking. And it is pulling along that vesicle. This vesicle is being moved from one end of the cell, perhaps, to the other end. And this is a protein that is walking, and the protein carries it from one end to the other end. So it walks along something like a railway track, which is actually nothing but actin filaments. There are also proteins that are able to push. These are there in the muscles. Actually, this is an example that I have. See, what you can do is you can take these proteins out of the system, out of your body or wherever they are, and then attach them to a glass slide. So these are the proteins. These green things are the proteins. They have been attached to a glass slide. And these objects that you see are actin filaments. So what you have done is you have attached the proteins to the glass slide, And then you have water, of course, and then inside the water you put actin filaments.
Myosin motors can be attached to the surface of a glass slide. Fluorescent actin filaments will bind to the motor domains of the attached myosins. When ATP is added, the myosin motors move the actin filaments. This rapid movement can be observed in a fluorescence microscope as the actin filaments appear to crawl across the slide.
So therefore there are nanomotors which are able to push. And this brings me to another very interesting example, the life cycle of a bacteriophage. As I have confessed in the beginning, my own knowledge of biology is very poor. I myself am interested in the theory of these things, and of course, you see, this is again another problem on which I have done some theoretical work. But as I said, my knowledge of biology is very poor. So let us look at a bacteriophage. What is a bacteriophage? Well, it is just a virus. Okay, this is a virus. What does it do? It infects a bacterium instead of infecting us human beings. So therefore, the bacteriophage, this is the virus. What is the structure of the virus? Well, it has what is referred to as a capsid. Well, it looks like a satellite. So it has what is referred to as a capsid, which is actually made of proteins. Inside that capsid could be a single DNA molecule inside this, or maybe an RNA, whatever it is. There is a molecule that is inside it. And then, of course, there are leg-like portions. And what the bacteriophage does is actually it goes and attaches to the cell membrane. The words that I have used are DNA, capsid, and the bacterium. And what I am going to show is actually the infection of a bacterium by a bacteriophage.
So let's start with the lytic cycle. The lytic cycle can be broken down into a series of stages, and these animations are really good. Notice on the picture, the virus is landing on these little bumps that are located on the outside of the cell. So the virus lands on the cell membrane, and you can see from the labeling of the picture, these little bumps are called receptors. And I like to use the analogy of a lock and a key. The receptor is kind of like the lock, and the virus like the key. If the key fits the receptor, we'll then proceed into step 2 of the lytic cycle.
Okay. Now there are 4 steps in this which I am going to show, but before I go into that, you see what is the interest of a chemist in this? Because as I said, I myself am a chemist. See, for example, if you look at this, the virus actually is able to recognize the surface and therefore attach. Well, how does it recognize? You see, it doesn't have a mind. So therefore, you see, there are chemicals that are there which it recognizes. So this is actually a chemical phenomenon. Viral recognition of the binding site is a chemical phenomenon. And how does it occur? Well, what kind of interaction is it? As a chemist, one can think of different kinds of interactions: electrostatic, hydrogen bonding, weak interactions, or even covalent bonding. So the chemist will be interested in understanding the process of this attachment. And in the next step, you will see that not only has it attached, these legs, they will actually bend so that the DNA can enter into the bacterium. You will see that.
Step 2 is called entry. And notice how the animation has changed. The virus is contracting, releasing its genome into the host cell, again, the genome being the DNA or the RNA. So what happens is once the virus attaches to the receptors, enzymes from the virus will help to weaken the cell membrane to the point where the virus can just contract and penetrate its outer body, its capsid, and then inject its.
Okay, this is what I was telling you. See, once it has attached, you see the things are not over. The legs actually kind of bend, and then a hole is made in the membrane. And then the DNA is actually injected. Now, Again, a chemist will ask, because I'm a physical chemist, I'm more interested in that kind of question. What causes the bending of the legs? Again, this is a chemical process. So what is the chemical process involved? What is the mechanics of this bending? And once the legs bend, you see the capsid gets closer to the membrane and the cell membrane, and the membrane is actually penetrated, right? The hole is made. How is that hole made? And once the hole is made, the DNA actually goes inside. Why does the DNA go inside? Why does it not sit inside the capsid itself? So these are all interesting questions, particularly of interest to a physical chemist. And let us look at the next one.
Step 3 of the cycle is called replication. And like the name implies, replication implies that more copies are going to be made. And you can see that happening in the animation. The virus DNA, if it's a DNA virus, will be used to make proteins. If it's virus RNA, because again, some viruses only have RNA in them, that RNA will be used as well to make proteins. So the DNA or the RNA of the virus will use the host cell's ribosomes to make.
Okay, yeah, well, what is happening is that the DNA of the virus enters into the cell, and then what it does is it makes use of the cell. You see, it makes use of the cell machinery to make proteins that it wants. So several large numbers of these proteins are made, and these proteins have the property that they will self-assemble. You remember self-assembly with respect to alpha hemolysin. Similarly, the proteins that the DNA of the virus makes, they will self-assemble, form a capsid, which is like this with the legs and so on. And then what happens, you see, you have a problem there because the DNA is actually outside the capsid and the capsid is empty. The DNA has to go inside. Somehow it manages to go inside.
Assembly. Those proteins are going to be assembled into new viruses. So what you see happening in the animation, again, you have in this case, 4 viruses being assembled. In reality, it's more than 4. The animation is just showing that how the process continues from replication into assembly. And once the viruses have been assembled, we come to step 5, known as release. The viruses will release enzymes that cause the cell membrane to.
Okay. Now, this is again another problem on which I have done some work. Again, I will not go into the details of my own work. Say, imagine you have the capsid, it is empty, and you have the double-stranded DNA. This is the double-stranded DNA, and the double-stranded DNA is actually outside the capsid. And the DNA, there is a problem here. The DNA has to go and sit inside the capsid. And the DNA has to get packaged. If you look at the final result of this, you will see that, well, the DNA is actually, this is the final result. The DNA is actually packed extremely well inside this capsid. Does this happen spontaneously? Would you expect it to happen spontaneously? No, it is impossible. It is impossible because the DNA, double-stranded DNA, is a rather rigid molecule. It would not like to bend. But if you look at this, you see there is very little space, so therefore the DNA has to be bent if it is to be arranged in this fashion. I shall give you the actual dimensions of this capsid in the next 1 or 2 slides. So if you had the empty capsid and if you had the DNA outside, the DNA would never go inside. If you can wait for as long as you want, you can wait till the end of the world, nothing is going to happen. The DNA will simply remain outside if you didn't do anything. So therefore, you see, there has to be some mechanism. There has to be some mechanism which actually causes the DNA to go inside. And in fact, in nature, there is the mechanism. What is that mechanism? It is a motor. So what am I talking about? Well, this is actually partway through the filling process. And what is happening is that at the mouth of the capsid, in this region, there is a motor. You will see the details of this motor in a few seconds. And what does it do? It pushes the DNA inside. This motor, how does it work? Well, it gets energy from ATP. ATP is hydrolyzed, and that hydrolysis causes this motor to rotate. And it is believed that this rotational motion somehow drives the double-stranded DNA inside. And this obviously is a nano-sized motor. So to give you an idea, well, this is actually what is known as the DNA packaging motor. It is an assembly of proteins. And this assembly is sitting at the mouth of the capsid. This is the capsid. It is sitting there. And you can see that the double-stranded DNA is passing through this. It is shown here as well as there. It is passing through this. And what is happening is that this assembly rotates. And you know that DNA is like a bolt. It has grooves. DNA has grooves. And therefore, the rotational motion of this actually drives the DNA, which is like a bolt. It is driven inside. That is what we believe. And let me give you an idea of the sizes of these things. The capsid is actually 54 nanometers in height and 45 nanometers in diameter. So for 540 angstroms and 450 angstroms are the height and width of this capsid. Now, if you think of the double-stranded DNA that is going inside, that is actually 6,600 nanometers low. So if you compare the sizes, you see the DNA is very, very, very long, and it is somewhat rigid. But it is going inside. Why does it go inside? Because there is a motor sitting there which takes hold of the DNA and pushes it inside. I mean, you think about it, this is really marvelous thing to happen at the nano level. And this is again one of the strongest known rotary motors. This is how it works. I will not go into the details of how the rotational motion, because this again is not very well understood. But that is the double-stranded DNA. Here is the protein assembly that rotates. And if you looked at the animation carefully, you will see that the double-stranded DNA is actually going inside. And essentially what is happening is that you have a nut and a bolt, and the nut is actually rotating. And if the nut is rotating, the bolt has to move. So therefore, the rotational motion of the protein assembly is actually converted into translational motion of the double-stranded DNA. And that translational motion actually pushes the DNA molecule inside. Now again, you see, we human beings are rather cruel in a sense, because we like to do very interesting experiments using these systems. So here is a double-stranded DNA that is in the process of going in. So that is the capsid, and this is the double-stranded DNA molecule that is going inside. These days, it is possible for you to design a single molecule experiment. What is the experiment? You take the hold of the capsid physically. How do you take hold of that? That is not very difficult, because you can attach it to a micro-sized bead, a plastic bead. You can attach it, and the DNA also you can attach to a micro-sized bead which is micrometers in dimension. And then it is possible for you to take hold of these two beads physically, well, using actually what is known as a laser trap. I will not go into the details of that. So therefore, this is attached to a micro-sized bead. The DNA is attached to another micro-sized bead. And then you can apply a force between these two beads, causing the DNA to come out if you want. The DNA which is going in, if you want, you can prevent it from going in by application of a force in this direction. This was an experiment that was done in 2001. In fact, you can measure the rate of this going in, rate of packaging as a function of this force, and there have been experiments doing that, reported in 2001. Well, these are all things that exist in nature, and these are all naturally occurring, but the question is, have people made nanomolecular motors? Okay, well, chemists, you see, because when we became aware of these things, it's only natural that we will try to imitate them. People have in fact tried, and I will give you some details of systems that people have made. But then before I go into that, let me tell you something. See, you can ask what is the efficiency of, there are man-made nano-sized motors available now, but what is the efficiency of these things? The answer is that they are not at all efficient. The naturally occurring machines, motors, they are efficient almost to the extent of unity, right? You almost 100% of the energy that you put in is converted. Well, it would not be exactly 100%, but almost 100% people say is converted into work, right? The efficiency of a man-made motor is extremely low. It may be, if the naturally occurring one has an efficiency of 1, the human-made ones would have an efficiency of probably 10 to the power of minus 10 or 10 to the power of minus 8 of that order. So they are extremely inefficient as of now. So what are the attempts I shall give you? Because you see I'm a chemist, I would like to give you some examples from chemistry. There's some attempts from chemistry, I'm going to show them to you. In fact, this was the work that I'm going to show you have been recognized with the award of a Nobel Prize in the something like 3-4 years ago. So this is one of the things that chemists have done. They have made it what are known as interlocked molecules. So here the idea is, you see, you have two molecules. One is, both are in the form of rings, one ring and another ring. And what you do is you interlock these two rings. So therefore you have such a system, but notice that there is no bond between these two rings. You see, this is one molecule, a single molecule, this is another molecule, and they are not bonded together. So you have interlocked rings. This itself is interesting. These molecules were made in 1983. These are known as catenanes. Important thing is there is no bond between these two rings. You can have another kind of interlocking. For example, you have a ring here, and you have a chain, a rod-like thing here, but the two ends of the rod are big. So therefore, you say this ring can actually move along this rod, but it cannot go out because it is actually prevented by the bigger end portions. So this is another example of interlocking. These molecules have been made and they are known as rotaxanes. They were made in 1991. Stoddard is a person who shared the Nobel Prize for this and other works. So if you are a chemist, I suppose there would be a few chemists in the audience. They would be interested in the structure. If they are not aware, this is the slightly more detailed chemical structure of this system. Now, the way the chemist made this is actually there is a ring and then there is this rod. But this rod has two portions, you see, which I have shown in different colors. This is one portion, and that is the other portion. And what you do is you make the ring and this portion in such a fashion that the ring likes to sit there. So therefore, if you have this molecule in solution, usually the ring will go and sit there. Okay. But then what you do is you change the chemical nature of this. You see, for example, what you can do is you can increase the hydrogen ion concentration in the system. And imagine a hydrogen ion goes and attaches to this site. Then the nature of that site is changed, as a result of which the ring is no longer comfortable sitting there. And then what it will do, it will move from there and go and sit there. So that is the kind of system that people have made. This ring, because it is moving from one region to the other, it is referred to as a shuttle. And this portion, because the ring is sitting there, it is referred to as a station. So these two are the stations and that is the shuttle. And this portion, of course, doesn't allow the ring to pass through, and therefore that is referred to as a stopper. So you have the addition of a proton or perhaps the removal of an electron from this station, then the chemical nature of the station changes, as a result of which the ring will go from here to there. So that is the kind of thing. People have actually made these things. You can have similar things happening in the catenanes. you have two rings interlocked with one another, you can actually cause chemically one ring to rotate. This is referred to as circumrotation, and you will see it as an animation here. You see one ring, you can cause it to chemically rotate with respect to the other. This has been achieved actually. And again, you may ask, what is the use? Well, the answer is that at the moment there is was of now no use, but it is interesting, and hopefully this will be made use of later on when we actually are able to build a more complex system. People also have made what is referred to as an elevator. So this is what is referred to as a molecular elevator. The Nobel Prize winner Stoddard made this in 2004. So if you look at this, you will see that there are two portions. You see, this is the portion which I will refer to as the lift, and this is the portion that actually moves with respect to the rest of the system. You will see that as an animation, I suppose. Yeah, okay. What you do is you change the chemical nature of this station. Then the elevator will, the lift will move from this point to the next point, and then you bring the chemical nature back to its original nature. Then the lift will come down. So that is how people have made, and the distance by which it moves is only 0.7 nanometers, so essentially 7 angstroms. Well, if there are chemists, they may be interested in more detailed structures. So this is the actual chemical structure of this molecular elevator. Feringa, who again shared the Nobel Prize, he made what is referred to as a molecular car. Well, here I have only an animation. This is a molecule that he has made. What he does is actually shoots electrons at this end, right? There is a technique called scanning tunneling microscopy, and so you have the scanning tunneling microscope which has a tip, and this tip is brought near this end, and it is used to shoot electrons at this, and when you shoot at it this using electrons, that portion rotates, resulting in the movement of the molecule on the surface on which it is sitting. So this is actually an example of what people have referred to as molecular car. So these are the things that human beings, that chemists have made, but as I told you, they don't have an efficiency close to the natural one. So it is really a challenge to make a nano-sized, artificially synthesize a nano-sized motor. So therefore, the question is actually, can the chemist make an efficient nano motor that uses chemical energy? And in fact, this is what I tell the students to whom I give the lecture. This is the IIT Palakkad view of the front portion of the part of the main campus actually. So this is at the moment, this is the main building, and you can see the IIT logo. And what I tell them is that, okay, maybe you will graduate from here, and then hopefully, well, I don't expect everybody to be interested in this, because you see, you have to be a chemist, you have to know biology, and you have to know physics. All these things you have to know, and hopefully at least some of them will study these things and perhaps synthesize in future sometime more efficient nano-sized motors. And that brings me to the end of my talk. I hope I was clear enough, and I would like to thank you all for your attention. Thank you, Doctor. Thank you, Father Binoy.
Thank you so much, Professor Sebastian, for this wonderful lecture. As the title sounds, it was really amazing. You have taken us into the world of the amazing world of nanotechnology, or nanometers, or nano dimensions. Well, you have explained even the complex concepts of nano pores, nano motors, and interlocked molecules. These are really complex, but explained in connection with examples from the nature as well as daily life. And what strikes me most is the interdisciplinarity of chemistry, how it's been applied in the contemporary times. Million thanks, Professor Dr. Sebastian. Now it's question hour. I request the participants to ask the questions directly, either ask directly or message. Accordingly, Professor Sebastian will respond. Kindly unmute your speaker and talk.
Sir, I will just say, Jacob Thomas.
Yes.
Further, I will just say that this is a type of lecture we will have in the LIPI series, because this is interdisciplinary, it touches on different subjects, and it's very, very lucid, it's very clear. And I'm sure it will motivate many of the youngsters for research, for explaining science to the common man. And I think Sebastian has done a wonderful job. I think we should give him a, I don't know whether if we clap, here or not, I am doing it here.
You did it. Okay.
Thank you. Thank you very much.
I see Babu Joseph, Dr. Babu Joseph in the video. Sir, you can speak. Dr. Babu Joseph, you can speak.
Professor Sebastian, happy to have you here on this platform.
Oh, it is a pleasure.
I couldn't interact with you earlier. I was trying to say something when your lecture was done, and also earlier when you identified me in the audience. In a better late than never. I have enjoyed, personally, I have enjoyed your presentation very much. But I was wondering, is nanotechnology equipped to apply the fruits of this kind of research today? Can you comment on that?
Yeah, well, you see, I have already given you an example where it is used. The Oxford Nanopore, they have made a DNA sequencer. It is costing only $500. You see, earlier when the human genome was sequenced, it involved so much effort. And if you have this particular equipment, it would be possible for you to sequence the DNA perhaps in a day. I mean, the human DNA, I mean, because you can have several of these machines running at the same time, and it will be possible. So therefore, this is a huge advance in technology, and the expense has come down so much. So that's probably the best example.
Yeah. This will, can we foresee applications in fighting viruses that cause harm, like the COVID-19 virus?
Yeah, well, you see, there is a book. There is a book.
Mechanism of infection is somewhat similar, isn't it? Mechanism.
That is true. Now, let me say something in here. You see, the way nanotechnology evolved, there was an origin. There's a book written by somebody called Eric Drexler. He was the one who was thinking of nanotechnology well ahead of many people. But then, of course, he was not a chemist. He was I think he was an engineer. And what he said is that, you see, you can make nano robots which will go inside the cell and perhaps catch hold of the virus and kill it.
Right.
So this is like a soldier going sent to an enemy territory with the purpose of killing an enemy soldier. And he does it. But you see, that is not going to happen. You cannot make a mechanical object which will go and identify an enemy and then kill it. That is simply impossible. The way you have to work is actually you have to make nano-sized things which will recognize your virus chemically and attach to it.
Let us hope it will happen.
Well, that is not easy, because you see the virus, you think of the COVID itself, you see people say that it is getting attached to your body. Now, what is that interaction responsible? What is the interaction responsible for that attachment? As a chemist, I would say, okay, this is known as hydrophobic interaction. It cannot be a chemical attachment, because if it is attached chemically, then it will never dissociate. It will be very difficult. Therefore, it has to be a hydrophobic interaction. And then people say, okay, if you had the COVID virus, if you touch it, it will go to your hand. So that means from the surface, it is transferred to the hand. Now that again, I mean, how does it take place? Well, it must be because the surface, the attachment to the surface is weaker than the attachment to your hand. Right? And then and then it says it goes into your mouth. You see, I find it somewhat difficult to believe, because see that if the attachment your hand is stronger, then it will stay on the hand. It will not detach and go inside your body. So these are all chemical interactions. You have to think about it very carefully. This,
But how is it possible that soap can kill the virus?
oh, soap is a detergent. You see, it will solubilize. You see, the thing is, there is the formation of measles. You know what measles are, I suppose. So what happens is that the soap solution, actually there are measles inside, and the virus is happy to be, I mean, not happy. It will be unhappy because it will not replicate. But physically speaking, you say that is a low free process that lowers the free energy. The soap, I am sure it is. What is happening is that the soap molecules will come and surround the virus, and the virus goes into solution. This must be the process that is happening, though there is no evidence. As a chemist, I can speculate. This must be what is happening.
I see.
The virus is removed because the soap molecules are actually sitting around it and solubilizing it. It is much the way the soap removes dirt. And once it is solubilized, you see, it would not be able to come back. So again, this is speculation, but as a physical chemist, I feel certain that this must be the process.
Right. Yeah, very interesting. Let us hope for better times.
Well, as of now, the only possibility is to have a vaccine. I mean, there is no other way as of now.
Yeah, thank you so much.
I think if that's the case, we will slowly wind up the session. Professor Sebastian, you have been talking to us, delivering the lecture for almost 1.5 hours to 1.5 hours. It's been really enriching, very insightful and informative, and this has widened the horizon of nano world just in front of us. And million thanks. And I know that today was a day of webinars for you and to the participants for your information. It's been just out of a comprehensive webinar online board prior to this meeting, and made it despite your hectic schedule. You have been so gracious to offer this lecture. This was a wonderful afternoon for us. Thank you so much, Professor Sebastian. As I mentioned, this will be YouTubed